Hello everyone. Griffith proposed the law of inheritance, and around the same time, Frederick Mitchell discovered a substance called nuclein, now known as DNA. Yet it took decades before scientists could prove what the genetic material really is. इतने समय तक जेनेटिक मटेरियल के बारे में क्लैरिटी क्यों नहीं थी? Because even though chromosomes were known to carry genetic traits, scientists did not know which molecule inside the chromosome did the job, DNA or protein. In 1928, the British bacteriologist Frederick Griffith conducted a series of experiments using Streptococcus pneumonia, the bacteria responsible for pneumonia. There were two strains, S strain, or also known as smooth or virulent, which is disease causing. Second is R strain, rough strain, non-virulent, or that did not cause disease. S strain has a polysaccharide mucus coat, making it smooth and dangerous. R strain lacked this coat, making it harmless. Griffith made a surprising discovery. In their experiment, he took live S strain and injected in the mice died. Then live R strain injected into the mice survived. Then he heat killed S strain and injected into the mice. Then mice survived. After this, he mixed the heat kill S strain with live R strain and injected into the mice. As a result, the mice died. Recovered from the dead mice are live S strain found. Griffith concluded something from dead S strain transformed the R strain into a virulent form. He called it the transforming principle. Though Griffith found transformation, he did not know what caused it. At this time, Avery-McLeod and McCarty, they isolated different molecules, DNA, RNA and proteins from heat kill S cells and tested which one caused the transformation of R into S strain. They inject DNA, RNA and proteins one by one into the mice and check out which molecule transformed from R strain into S strain. The result was only DNA caused the transformation. They used enzymes to confirm it. They used DNAs that destroy the DNA and no transformation. Only DNA could cause the transformation. They use enzymes to confirm. They use protease to destroy protein. Then the result is transformation occurred. After that, they use RNAs that destroy the RNA. Transformation also occurred. And then they destroy DNA. When they add DNAs, no transformation occurs. It means that DNAs that destroy DNA and without DNA, no transformation occurs. This strongly suggested that DNA is the genetic material, not the protein or RNA. But some scientists remain skeptical. The conclusive evidence came from Alfred Hershey and Martha Chase using a virus that infects bacteria, the bacteriophage. Let's break it down. The bacteriophage has two parts, protein coat and DNA coat. They wanted to find out which part enters the bacterial cell during infection. They designed a clever experiment using radioactive isotopes P32 phosphorus, radioactive phosphorus, to label DNA, since DNA contains phosphorus, and radioactive sulfur S35 to label proteins, since प्रोटीन्स कंटेन सल्फर। आपके माइंड में ये क्वेश्चन आ रहा होगा कि उन्होंने बैक्टीरिया को कैसे सेपरेट किया। दे लेट द रेडियोएक्टिव वायरसेस इन्फेक्ट ई कोलाई बैक्टीरिया, देन यूज ए ब्लेंडर टू शेक ऑफ वायरल पोर्स and a centrifuge to separate the bacteria from virus parts. In the experiment, he finally concluded that virus with P32 DNA, and after checking radioactivity inside the bacteria, and virus with S35, the radioactive sulfur, there is no radioactivity inside the bacteria. DNA entered the bacterial cell and carried genetic instructions. Protein did not. इस एक्सपेरिमेंट ने यह बात पूरी तरह से क्लियर कर दी कि डीएनए ही असली जेनेटिक मटेरियल है। दिस वाज द फाइनल प्रूफ दैट सेटल द डिबेट। लेट्स रिकैप द जर्नी टू डिस्कवरिंग द जेनेटिक मटेरियल। Griffith experiment in 1928 discovered transformation. Avery-McLeod-McCarty in 1944 identified DNA as the transforming substance. Hershey and Chase 1952 proved that DNA is the hereditary material. आज हमें obvious लगता है कि DNA जेनेटिक मटेरियल है, लेकिन तब ये कितना बड़ा ब्रेक थ्रू रहा होगा। Yes, it was a milestone that gave rise to modern molecular biology.